Hello everyone, welcome to the biological database video. If you're a student or a teacher who's completely new to bioinformatics, consider this tutorial as a very basic guide or an explanation to the biological database. Hello everyone, Sanket here. Today we are going to look at a biological database which is called as GenBank. GenBank is a part of NCBI, which is a part of NIH. NCBI, as you know, stands for National Center for Biotechnology Information, whereas NIH stands for National Institute of Health. So today we are going to explore how the GenBank works. Right. So first, when you are at Google, you type GenBank. So then you see the first hit, which is there of GenBank overview. Yeah. So this is how your homepage for the GenBank actually looks like. You can see that there is a drop down here which says it is a nucleotide. So GenBank is actually a nucleotide database, and that is why all the information which is present in the GenBank is related to the nucleotide. So that means your DNA, the information or the sequences which is related to RNA will be present in your GenBank, which is there. All right. The interesting thing about GenBank is when the Human Genome Project was actually completed, all the data which is related to the Human Genome Project was incorporated in this GenBank database. So you can see the search here, and then you can search for the various sequences basically in your GenBank. So let's say if I type cancer. So I can I can search the different sequences based on any diseases also. So if I type cancer and then I click on search, so you can see the hits. So 10 million hits for cancer, and all of these, okay, are nothing but your sequences. Okay, the various types of sequences, which right. What is more interesting is what is there on your left hand side. Okay, so now these are the filters. So how does GenBank actually work? Suppose you develop a sequence in your laboratory and then you want to deposit that sequence. You deposit the sequence in NCBI. All right. And if it is a nucleotide sequence, then it becomes a part of this GenBank. So all these sequences, what you see here, all right, they are either derived. So they are derived from the experimental studies which are done by the various scientists. Now, they can be done in different categories. So, like you have the species here. So you have animals, plants, fungi, protists. So these are the cancer sequences which are present for the various species. So there are about 8 million which is there for animals, about 581,000 for plants. So like this is the various cancer sequences which is present for the species. You can here you can try and understand that what all sequences are there. You can have genomic DNAs and RNAs, you can have mRNAs, you can have rRNAs. Here you have the resource databases. So it's the GenBank itself, and ref sequence is nothing but your reference sequences. Okay, that is what this database stands for. These both are the name of the databases. GenBank is what we're actually looking at. You can have the sequence type, that is the nucleotides, which as I said is DNA and RNA, or you can have the EST or the expressed sequence tags. Okay, so EST stands for express sequence tags, or you can have GSS, that is genome survey sequences. So that means these are GSS contains the information of the entire genomes which is present. You can have the cancer sequences which is present in chloroplast, in mitochondria, plasmid, right? And then you can develop a custom range for the sequences. So suppose it could be something like you want from 100, all right? Yeah, sorry, one from 100 to, let's say, 1000. Okay. This I'm just doing it randomly. Okay, if you have a specific sequence in your mind which you're working on, all right, and you know its range, it will be very easy for you to find out. So if I click on apply, Then you have the release dates, then you have the revision dates. Okay, so when is the sequence got released, based on the year, everything will be done. So yeah, if you see, I had clicked on apply. Now all the sequences which are obtained here in front of me are the ones which are between 100 to 1000. Okay, so that's because I had applied the filter. On my right hand side, which is here, is nothing but results by taxon. Okay, so hopefully you understood what the left hand side is. Okay, here it is nothing but the filters which you can actually apply. Similarly, you have another filter which is present on the right hand side, which is called as results by taxon. Okay, so like you have the species here. Okay, and we had, I think, selected animals here. So in this you can have the different organism which is there. Okay, you want human, so this numbers are the number of sequences which is present. All right, so you want to find out the all the cancer sequences which is present in homo sapiens or in bos taurus, okay, or in your rats, in your mice. All right, so let us do it that we want to actually analyze. So I'll apply filter here. Okay, I want all the cancer sequences from 100 to 1000 sequence length, okay, for homo sapiens. All right, so I clicked on Homo sapiens here. So now all the sequences which I have, okay, you remember there were about 6 million sequences or something. Now it has come to half, which is 3 million. So now all the sequences which is there in front of me is of only and only Homo sapiens, with the sequence length from 100 to 1000. Okay, so this is how you can actually explore your GenBank. Okay, you can search it using a specific disease. You can apply filters based on what you want. I have not done a lot of filters, I just applied it on once, but if you have a more information or knowledge about what sequences you're searching, you can apply more filters. Okay, this is where your search details are. So this is where your filters which you have applied can be seen. You can see the search details here. All right, so now let's actually jump into how the GenBank actually looks like. So let me click one of the hit. Let's go for the first one itself. Okay, Homo sapiens keratin associated protein 32 K KR type mRNA. Right. So let me just click on this. All right. When I click on it, right. So this, my dear friends, is how the GenBank actually looks like. Okay, so yeah, so this is the title, all right, which is your Homo sapiens keratin. This, if you can see, is NCBI reference sequence. All right, now this, my dear friends, is NM underscore. This is nothing but your accession number. Okay, so like in your colleges you have your roll numbers. Similarly, in your similarly, each and every sequence which is being uploaded in this database has its own roll numbers which is there. Okay, roll numbers or enrollment numbers or so whatever. So we'll call this term which is called as an accession number. All right, then what you have is all the information which is related to this. So you can have locus, which is nm, that is here is your accession number. 714 base pairs, that means the number of residues which is present in the sequence is 714. It's an mRNA sequence, it's a linear. Okay, so its shape is basically linear. PRI stands for primates. Okay, because we have taken Homo sapiens, so here it will be PRI, which is primates. Now depending on what species are you using, this thing will change. Okay, it was uploaded on 25th of July 2020, so it's a pretty recent sequence which has been uploaded. Definition is the name of the sequence here. Again, this is your accession number, this is the version. Now what does this version mean? If you can see here, it's given 0.3. That means this Homo sapiens keratin associated protein 32 or KRTAP2, okay, has been updated. This sequence has been updated multiple times. Now this is the third version of it. So initially there was one sequence which was submitted. Then somebody found out another same sequence, but then I think it had much better, much better results than which was there in the first version. So then there must be a point 2, and now this is point 3. So like this, you have versions here. Okay, so the versions here stands for the number of times it has been updated. Then you have the keywords, which is your reference sequence, which is there. The source is Homo sapiens, which is human. So you have eukaryotes, metazoas, and all, right? Yeah. Then you have the references. Then you have the authors. So these are the authors who had actually worked on. So all this thing is a list of the authors who had worked on finding this sequence. We have the title of the paper. The paper was published in this journal called as Nature. All right. This is your PubMed ID. All right. So if you click on PubMed ID, all right. So I just clicked on this PubMed ID. It will take me to another window, which is your PubMed window. All right. I won't talk about PubMed window here because there's another video where in which I'll be discussing more about PubMed. All right. So just to show you, this is how the PubMed thing looks like. Right. So this is your PubMed ID. Then you have the references, then you have the authors, then again the title. Okay, now this is basically the another paper in which the work had been done. So if you can see, this is another PubMed, this is another PubMed ID. So this is one paper, this is second paper, this is third paper, this is fourth paper. All right, this is another fifth paper, I think. Oh, sixth paper. So this means that this a lot of work, okay, has been done on this protein, and number of research papers have actually been published. All right. But then every time someone publishes a paper, there has been some updates on the sequence. When I'm saying updates on the sequence, that means the first time when the sequence was actually found out or developed, there must have been some issues, some nucleotides must be missing or something of that sort. Well, now you'll be asking, why is that so? This does happen. All right, this is all experimental techniques. So whenever you are finding out sequences experimentally, there are chances, okay, where you can have such kind of issues being risen. Okay, yeah, that's why there are some problems where it happens with the sequences. But then as you keep on working on those sequences, you get a much better and a cleaner version of those sequences. All right. Then you have the features. Okay. So the features, you have the source, you have the gene, the exon. So gene is this KR trap belongs to what gene? It belongs to what exon sequence? There is also CDS, which is the conserved domain sequence, and these are the miscellaneous features. And the final thing is your actual sequence. All right. So you can see from the first to this is just 661, but then it is 714, which is what it's all about. So this is a total 714 sequences. Okay. These are this is the ladder. Okay, this is for your reference that which sequences line it is actually. All right. Have the initial headers which contains all this information. Then you have the middle section, okay, which is nothing but your sorry, you have the middle section here, which is nothing but your features, okay, and then you have a third section which is your sequences. All right, all of these are links. So if you click on any of these links, it will take you to that particular thing. So in this case, let me just click on the gene here. And if I click on the gene, this if you see the highlighted thing, this means this entire sequences of 700 is basically a gene by itself. What happens is sometimes you have a entire huge genome, and when you click on gene, only a particular section is highlighted, right? Now this is anyway a very small sequence, and it is completely a gene by itself. So that's why you'll not get to know what I'm saying. But if you have a huge sequence, about 10,000 sequences, and you want to understand what is genes are, when you click on the gene, only the gene section gets highlighted. So then you don't have to sit and find out where the rest of the parts are. It becomes very clear and easy for us to understand and analyze. All right. Yeah. The next thing is these two parts. Okay. So if you see, there's something which is called as FASTA and something which is called as graphics. So let's click on FASTA here and understand. So click on FASTA. So if you see, the sequence for the FASTA will get uploaded. This is, my dear friends, your FASTA sequence. Okay. Now, so this is your entire sequence from GTA to GGCA. This is your sequence. And what you see in the starting is nothing but your faster line. This is very important. This actually makes it as a faster sequence. If there was no line and it was only the sequence, it would have been called as the plain sequence. But because I have this line, that's why it's called as a faster sequence. Now, FASTA is basically a program. It stands for fast alignment. It is a heuristic alignment method for sequence alignment. And this FASTA program or the tool had its own set of sequences, and that's why those sequences are what we call as the FASTA sequences. All right. How does the FASTA sequence look like? It starts with a greater than sign, as you can see here. Okay. Then it has the accession number, which we were already discussing about, and then it has the description. Okay. And finally, it has the thing which is an mRNA or what. So you have the greater than sign, you have the accession number, then you have the entire description, and then finally you have the a sequence. So this entire things becomes your FASTA sequence. The reason for explaining you this FASTA sequence is most of the softwares which is there in bioinformatics, they consider FASTA sequences for their analysis purpose. So if you're doing something like multiple sequence alignment, or you're doing something like pairwise sequence alignment, so all the tools which do all of these pairwise or multiple, they require this FASTA sequence. The next thing is the graphics here. Okay, so let's click on the graphics. Okay, I'll go back so that it will be very easy for you to understand. Okay, if I click on graphics, let's understand what I see in graphics. So as the name suggests, it's called as graphics. So it will give me the graphical representation of this entire sequence, what is there. Now, if you see, this is your gene, which is your KR TAP32. If you can, I don't know if you can see probably, but this green color bar, what you see is nothing but the KR TAP2 gene, the exact gene what we are actually discussing right now here. All right. And within this, there are three different sections. Okay. So you have the NP114 section, which is the red section here. Yeah, if I keep my mouse on this, yeah, so you can see it is the keratin associated protein 32. All right, and its location is on 54. So this is a small section which is there in KR tab. Then you have another thing which is called as keratin matx, and then you have another repetitive region. Okay, can you see this repetitive region? All right, so this gives you a graphical representation of your entire gene, how it looks like, what are the subsections. Now in this there are only 2 or 3 sections. You will see some examples, okay, where there are multiple sections in this, and it becomes very clear for you to analyze the entire gene and what are the regions which are present in this gene. Okay, so this is your graphical representation. Okay, so you can see the full view, or you can see the GenBank record of this region here. Okay, using the tools you can now it's up to you. You want to just have a look at the text view. You can also create markers, okay, for your understanding. Okay, like at what position in this entire gene you want to keep. So suppose it has 358. Okay, and I click on OK. So at the 350th position there is a marker. Now this is only based on how what exactly you are trying to analyze in this protein. Okay, I just put this randomly, but if you know your sequence, you know what you're trying to find in the sequence, what specific region you're trying to find in the sequence, you can apply markers to it for your analysis or for explanation of this gene, what is, right? And finally you can download this entire files or the FASTA format or what. So we looked at 3 things in GenBank. We looked at first is how the GenBank format looks like. Okay, what is the accession number, what are the 3 regions, that is your headers, features and sequences, which is there in GenBank. Then we saw what is a FASTA sequence, how does the FASTA sequence look like, what is the significance of all of these things in the FASTA. And finally, we saw how the graphics of the GenBank looks like. Well, it's not just always the name of the disease. You can search for nucleotide sequences by their genes. or by applying filters by their accession numbers, or even you can search them by their authors. So this was a beginner's guide tutorial for a biological database which is called as GenBank. Uh, Hopefully you have understood it. So